All right, hey guys, this is our first Zoom lecture, and what we're going to do today is we're going to go through the PowerPoint that I posted last week for 9 and 10. I'm very well aware that y'all are just scooting right through those PowerPoints, doing the 5 vocabulary that I asked you to do, and honestly, 9 and 10 is a really hard unit, so I want to make sure that we go through it together, and then you can get a little bit better understanding of what you need to know for this unit. So I also have posted a follow along worksheet on Edmodo, and as I go through this presentation, I'm really just going to scoot through this presentation, y'all, but I want to stop and hit major points that vocats are going to cover. We're going to continue to go through this with the assumption that vocats are still happening, because at this point, I don't know, and it's my job to prepare y'all as much as possible. Also, side note, I did do my hair for y'all today, so lucky duckies. Okay, so here are the things that I want you to know. When I'm done with this, I want you to have a complete understanding of the purpose of an integrated pest management plan. I want you to understand benefits and concerns of dealing with plants in biotechnology. Like, we can go over 100 benefits, pretty much common sense, but there are concerns with plant science in biotech, and I want you to have a better perspective of how micropropagation is beneficial to agriculture and biotechnology. So let's just keep scooting right through. Integrated pest management, programs of observation and calculation used to maximize pest control while minimizing both damage and the use of chemicals. The whole point of an IPM is to reduce the use of chemicals. That is the whole goal. That's what we're shooting for when we do an IPM, okay? Cooperative extension agencies nationwide will go out to farms, help farmers with IPMs. We want to limit pesticide damage to the environment because, as you know, we do have a tendency to get blamed for environmental damages. We want to scout, as observation allows treatment to proceed prior to a full-blown infestation. I have an uncle who this is like his whole job, is he goes out to fields that have a pest problem, and he just walks through the fields, figures out the pest, figures out IPM for farmers, and helps them implement them. It's actually a pretty cool job if you love bugs and whatnot, which I don't. It increases the effectiveness of all types of controls. So there are different types of non-chemical treatments. The first being a biological control. This is where you're going to utilize naturally occurring compounds and substances to control plant pathogens. Bio meaning life. Okay, so we're going to use living compounds to help control pests. Often more expensive, difficult to find, and it's requiring more frequent application than a chemical method. In recent years, new research and availability, however, have led to an increased use in both commercial and residential, and increased the value of the crops when used in a place of a chemical. So that's kind of like the same concept of people who use organic chemicals, organic control. Their products are going to be marketed at a higher price because people are going to be more interested in buying an environmentally safe product. Going back to biological controls real fast. A really extreme example, but an example would be, let's say you have 2 acres of property out in the middle of the country and it's tick season. You can spray your yard with a chemical to help control the tick population, or you could buy 10 guinea hens and chickens, and the guinea hens and the chickens will eat the ticks and reduce your tick population in your yard. That's a really extreme example. Plus, the guinea hens are really dumb. But that would be an example of a biological control over a chemical. Remember, the whole point of an IPM is to reduce chemical use. Chemical controls center on the use of pesticides, herbicides, insecticides, miticides, fungicides, blah, 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 used to kill plant pests. Most current insecticides will attack the nervous system of the plant so that when the bug eats it, you know, but it also can attack humans. So a lot of all farmers in the United States, in order to apply chemicals or pesticides, have to be licensed and certified in it once a year. So they have to go through the extension service or like a local pesticide service to get their yearly certification in order to put those things on their crops. And there's also the buildup and transfer of chemicals to the environment. Chemicals are synthetically manufactured in a variety of forms. So you can do dust, powders, liquid, foam, granule. I have also seen it in like seashell form, which is really odd, but we use that some on the chicken farms where I worked. Systematic pesticides enter the plant and then are transferred to all the plant tissue so that when the bug eats it, bye-bye bug, no more. Usually kill either by contact or ingesting. So here's 7-Dust, here's Roundup. I know y'all have heard of Roundup. Okay, I know you have. It's also like a huge lawsuit pesticide being attacked right now. And you have physical control. That's literally walking through the greenhouse, walking through your garden, and removing things physically from plants or capturing, so you can do traps, things to that extent. It is often the most cost-effective and environmentally friendly, but it is extremely time-consuming and slow. But it is an option. Rarely totally effective at destroying all pests. So imagine you are a student at NC State and it is your whole job to work inside the greenhouses and physically remove aphids from research tobacco plants. By the way, that's a real thing, and I know lots of people who did that. That would be horrible. I was not a plant person. I did animal science. So traps, cost-effective, environmentally friendly. You got that yellow sticky tape. I think I have some in my lab with the guinea pigs and gnats, but it can capture whiteflies and other small insects. Japanese beetle traps. So here's just a couple pictures of what you might see inside greenhouses, barns, etc., etc. Implementing an IPM should follow all the guidelines. Plan should include as little dependence on harmful chemicals as possible. Again, the goal of an IPM is to reduce the use of chemicals. Whole purpose of it. IPMs do utilize some chemicals, but we're trying to reduce. Utilize varying and responsive methods of control. Most important part of an IPM behind scouting helps to ensure that the overuse of one method of control does not produce insect resistance. One reason for the use of buffer zones around BT crops, resistant insects breed in non-resistant insects, preventing the development of resistant strains. So this is kind of what you've heard has happened with like the honeybee crisis. They think that because of pesticides that we've used, and we've used just one type of pesticide for so long, that we have actually killed out the bee population, which that's very much a possibility, but I really don't think they can pinpoint it on just one thing. The use of parasitic nematodes to control beetle larvae would be an example of an integrated pest management biological control. Physical control would be, again, sticky tape. And then the insecticidal soap sprayed over insects. This is actually a chemical control. But here's an organic fungicide that's still considered a chemical control, y'all. Okay, on that worksheet, that's on Edmodo. You're going to have stop session 1, stop session 2, where you need to put the answers. So the question for stop session 1, what is one example of a biological control? Bio meaning life, we're using life to control pests. How does that differ from a physical control? You can pause the video if you want, or we can just continue on, and you guys go and answer those. Jot this down somewhere, come back to it. So we're going to go over environmental benefits, environmental concerns, economical benefits, economical concerns of plants in biotechnology. And environmental benefits would be insect-resistant crops, the reduction of strong sprays, it's more beneficial, insects spared, it's a safer working condition, herbicide-resistant crops, reduced use of herbicides, reduction of soil erosion, things like that. Environmental concerns would be that resistance to BT. BT meaning, I feel like dwarf these. Okay, if you don't know what BT means by now, we have further fish. Reduced interest in sustainable agriculture would be an environmental concern, and transgenic plants becoming weeds. Transgenic is another word for GMO. So now we're worried about GMOs becoming weeds in the environment, which is pretty interesting. Pollution of native sources of new plant genes. Economic benefits, higher and more predictable yields with these IPMs, with these genetically modified plants. We know if we plant an acre of genetically modified corn, we're going to get a higher yield because we've genetically modified it for that. Market for new agricultural products. Hey, we have this new GMO, and this is the things that it's going to bring you. It's going to bring you a higher yield. Benefit to world food supply. Okay, now we've made a genetically modified corn crop. We know if we plant that we're going to get 98% predictable yield out of that field as opposed to 85. Now we're making more food. We have a better food supply for the world. And pharmaceuticals, safe and inexpensive to produce. If we can mass produce these things, the more of something you have, the less the price. It's simple supply and demand. But we do have economic concerns. We're going to shift towards larger and more capital intensive farming. Folks, we have lost the small farmer, and it's because of these economical concerns. It's because of these larger industries. The seeds for these GMOs are super duper expensive, and the smaller farmer just cannot afford it. And larger corporations, again, we're seeing it all the time. I know we're talking about plant science right now, but like larger corporations are taking out the smaller farmer in the way of, look at Walmart. They no longer buy their milk from small local farmers. They literally opened their very own chain of great value dairies. And when they did that in the United States, the dairy farms like reduced by like 15%. So there is economic concerns there, and the reduction of farmers' rights to save the seeds. If you were buying that genetically modified seed for that corn that's going to give you a 95 to 98% yield coming out of 1 acre, and you're buying your seeds from Bayer Crop, you can't keep that seed. You don't own that seed. Bayer Crop owns that seed. So yeah, you're planting it, you're getting that yield, but you don't have the right to replant that seed. The next year, you have to come back and you have to spend that expensive price right back on that genetically modified plant if you want to use it. So that's another economic concern. We have benefits to biotech foods. These are pretty much common sense. Higher protein levels, better tasting fruit, higher level of vitamins, reduced allergens, healthier food, lower fat. But we also have some concerns. Those hidden allergens, increase to natural toxins, unknown substances, labeling, yada, yada, yada. So here's our stop session number 2. What is one example of an economic concern for biotechnology and plant science? It's just a few slides back. So if you want to pause the video to do the worksheet, please do so. And that is an emoji of me. with a cat, making it rain. You're welcome. I appreciate you. Okay, biotechnology and pest control. What is BT? Bacillus thuringiensis is a soil-dwelling bacterium. If you do not know what BT is by now, we have bigger problems. The BT gene is used to provide plants with systematic resistance to chewing insects. Remember, they eat the plant that has the BT gene. It gets into their system, and the poor little things just die. Like this. Look at that poor little worm. The pest dies when feeding on any plant part. RIP. RIP. Okay, herbicide-resistant plants that are not affected by the use of certain systematic herbicides, and virus-resistant plants, few instances of successful implementation. So we're not moving very far when it comes to virus-resistant plants. Why? Back to Biotech 1. Viruses mutate very quickly. It's harder to kill a virus than a bacteria. Hello. Don't we know that? You're in the middle of COVID-19, y'all. So usually functions by creating a protein coat around the entire virus molecule. Approval for field trials with transgenic organisms, they have to be sought from APHIS. This is a VOCATS question. Most often required a significant amount of information produced. APHIS is who approves field trials for transgenic organisms. That is a VOCATS question. Agencies involved in regulations and monitoring would be USDA, EPA, and APHIS. APHIS stands for Animal Plant Health Inspection Service. Okay, we're almost done. Micropropagation in plants. This is a lot of really weird information. So as I'm cruising through this, I will tell you what is important for VOCATS, and let's just scoot through. Micropropagation is the growth and development of differentiated plants involving the development of cells from undifferentiated to differentiated states. AKA, we're going to grow plants in a lab. At Bayer Crop, 5 miles down the road from Jordan High School, this is how they grow all of their research plants. It's super interesting. Unfortunately, they no longer let us into their facility, which kind of stinks because I'd really like y'all to see it. Uses of micropropagation, it's used to create transgenic first-generation plants. It's used in horticulture to produce orchids, African violets, lilies, and ferns. And it's used in nurseries to grow fruit trees, evergreens, roses, and shade trees. Sounds so pleasant. Many genetically identical plants can be created from one parent plant. So that's propagation. We learned about that in Biotech 1. If you cut off a piece of a plant, you can actually dip that into a hormone, put it in some soil, and an identical plant grows. That's all we're talking about here. A little bit of tissue culture. Allows many plants to grow in a small place in a short amount of time. So we can cut 5 to 6 weeks off of a plant growing process. So we can speed up the plant science process. In some species, this method will produce a healthier plant. Good to know. We already know what plant tissue culture is, y'all. To propagate whole plants from a very small amount of tissue. Down here, there's a picture of them taking small tissue of plants, putting it in test tubes, and growing a whole new plant. That's all we're talking about. It's going to get really confusing. All we're talking about is growing plants from small pieces of other plants in an in vitro or sterile situation. Okay, plant cells used in this method may be taken directly from host plants or transformed through genetic engineering. Media, what we're talking about here is agar. What we are going to grow the plants in, the different combination of nutrients and growth regulators used to sustain the cell or the tissues. Media must be sterile at all times, and as the plant grows, as the plant cells grow, they are transferred into different mediums. So this is an important part. As I'm going through these 4 stages with you, I'm going to point out the important timelines and growth mediums for each stage. So let's go to stage 1. establishment of an aseptic culture, sterile vessel and sterile plant cells. This stage lasts about 3 to 5 weeks. That is important. Stage 1, 3 to 5 weeks. Establishment in an aseptic culture, and it is initiated from a small piece of plant tissue. That's what you need to know for stage 1. Okay, here's a picture of a plant in stage 1. Now we're going to go to stage 2. So stage 1, 3 to 5 weeks, establishment of aseptic culture. That's what you need to know. Stage 2, at the end of stage 1, we're going to take that little plantlet, this little dudette right here, okay, and we're going to reculture it to a new medium. We're hoping in stage 2 to get more shoots. Okay, this stage is going to be about 5 to 7 weeks. Still, your medium is sterile. And here's some of the things that can be inside that medium. Okay, you want some organic compounds, some vitamins, and some sugars. Here is stage 2. See the little shoots? Little shoots growing? Stage 2, you get shoots. Okay? Stage 2, 5 to 7 weeks. We're trying to get some shoots. There's stage 2. Let's go to stage 3. When those little plantlets have fully developed clusters of shoots, we're going to put them into another medium that will last about 6 weeks. That's what we get. Okay? We got bigger shoots. Because stage 4 is when we get roots. So stage 4, roots. Stage 3, big shoots. Stage 2 is when we get the little itty bitty shoots coming up from the plantlet that is in stage 1. Okay? We're going to be planting in soil, which is a non-sterile environment. And this lasts about 3 to 4 weeks. Folks, your vocab questions are going to ask you timelines. Okay, stage 4, shoots and roots for 3 to 4 weeks. And that's what you get. Okay, let's go back. Sorry, just gonna. Stage 1, itty bitty little plantlet in an itty bitty test tube. Okay, stage 2, stage 3, stage 4. Okay. Method of micropropagation is preparing the plant medium for culture, selecting the right material, preparing plant medium for culture. This is literally just talking about making agar, how to sterilize it properly, how to sterilize the plant properly, how to sterilize the water properly. Got to swirl that material in bleach for like 15 minutes, remove harmful remnants from the bleach, store momentarily in a petri dish, prepare agar for media use. Agar must be remelted in a container to shape. We all know agar just solidifies when it's cooled off. It's liquid form when it's warmed up. Y'all know what agar is. This is just telling you how to put the plant tissue on the medium. Okay, so place the tissue firmly on the agar. Tissue might be placed relatively close to the agar. Place tissue on the medium. All of this is just craziness. It's not really. Transfer tissue. Oh wait, here we go. Agar high in cytokinins is used to promote shoot growth. And then auxins promotes root growth. You guys learned that in Unit 9. Auxins for roots. Cytokinins for shoots. The way I remember that, those are both hormones, growth hormones. The way I remember that is the X in auxins kind of looks like roots of a plant. So that's the best way I can describe shoot growth and root growth to you. I know this has been a long video, and I'm sorry. But you'll need to take that worksheet and you need to fill it out with the video If you have any questions, please email me. My office hours are from 11 to 1 every single day. Y'all, I love you. This is a learning process. You're not in it alone. Look at me, I'm a big floating head. It's a Saturday afternoon. My kids are napping. We're all just doing the best we can, but you really, really, really got to try to stay and keep up with your work. Okay, I love y'all. I will talk to you later. And thank you for being so patient with me while I'm trying to learn how to be a distance education teacher. Bye, y'all.